you were using.
Myself is Ravindra Makde, and thank you, Dr. Dongarwar, for your kind introduction, and thank you for organizing committee for giving me opportunity to say, talk about one of the subject in method biophysics called macromolecular crystallograph. So we all know biological macromolecules like these. Am I audible?
Yes, sir, very much. You are audible.
Yes. Yeah. So these are proteins. Proteins can be glycosylated. These are also macromolecules, DNA, RNA, and then there are protein, DNA, protein, RNA complexes. These all comes under the biological macromolecules. They have a huge number of atoms, and their structures are very important for the function of biological cell. And to know the function, one has to understand the structures, so how they look. To give an example, the DNA structures was so instrumental in uh, putting forward the genetic code hypothesis. For example, when they determine the DNA structures, they actually proposed the model of double helix DNA based on fiber diffraction data, where you get some periodicity of the DNA. Based on that, one could infer a model of genetic code. One could infer the model of replications. One could infer the transcription and lot of things the structures of DNA has given. Same thing, the protein structures also has given inputs in the function of the proteins. For example, hemoglobin, how it carries the oxygen and how it carries back the carbon dioxide, and different other important protein structures. So as I told you, the functioning, understanding at molecular level is very important. Then one can assign the function to macromolecules. Suppose one macromolecule is there and there is no function is known. Based on their structures, their homology is known with respect to other proteins, and one can infer and assign some function to the macromolecules and what they might be doing at the cellular level. Then one of the most important is a rational drug discovery. You have all seen the period of COVID epidemic, and there the medicine for the SARS-CoV-2 has come from the one of the protein M-Pro peptidase. So they have found one inhibitor which bind very strongly to this peptidase and stop the activity of this peptidase. As a result, virus cannot replicate. And this was only possible because we could solve the structure of this M-Pro peptidase. This is the picture below is shown that a drug bound in the active site of M-Pro peptidase, and the right hand picture is showing the electron density, like where you can feed the drug molecules. So then the further affinity improvement of this drug can be done using this structure. So they can add some functional group into this drug molecule to make it more stable, more accessible, or pharmacologically more conducive. And as a result, finally they come out with a drug that is in the under trial, and in some country they have been used. Then other application of the macromolecular structure is a protein engineering. There are many proteins which are used in the industries and therapeutic diagnostic purpose, but they are unstable. They cannot stay in the room temperature for a long time. They have to be engineered, and this engineering of the protein is possible once we know the structures. So that's why getting the macromolecular structures of especially proteins. Proteins are huge because in human cells, there are estimated to be around 25,000 different proteins. And if you count their modifications and their post-translational um, modification, if we consider, then it comes down to 1 lakh different types of macromolecules. Then different types of RNAs. DNA has typically one structures, but DNA can also adopt some nucleosome structures. They can adopt some quadruplex structures. So you know, so getting the structures, seeing the structures is the first step to start with the understanding the molecules at the molecular level. So what are the different methods for structural determinations? These are very popular. Nowadays, the method called cryoelectron microscopy is becoming a very powerful method where you prepare a protein samples or protein complexes. It can be very big, it can be as good as a whole virus. You can freeze them in the liquid ethane and get the transmission electron micrograph of this frozen this thing, frozen structures. As a result, by then combining this image in the 2D form, one can derive the three-dimensional structures of them. This has revolutionized, and 2017 Nobel Prize has gone to something who has invested the cryoelectron microscopy. Other technique to get the high resolution structures is X-ray diffraction. X-ray diffraction by far is the first choice of determining the structures. It is the most easy method, and there is no size limit for determining the structure by X-ray crystallography. But the problem is one need to get the crystal which diffracts, diffract in the X-rays, and if that is possible, then you can get the structures of any macromolecules. So other is NMR. NMR is a structure of protein one can determine. So what is the problem with the NMR? That one cannot determine the bigger structures, like more than 25,000 Dalton structures. But unfortunately, most of the proteins are present in a complex form. They are very big in the humans, and their size many times exceeds 25,000 Daltons. But nevertheless, for the small molecules, small biological macromolecules, NMR is a very powerful tool, and it can study the protein folding, protein denaturations, protein modification. This can be very powerfully studied by the nuclear magnetic resonance called NMR. Then this year, based on the artificial intelligence, Google DeepMind has made a program which can predict the structure of the protein with very accuracy from the amino acid polypeptide sequence, just based on the computation. No, you don't require experimental approach, and their accuracy of providing the structures is getting improved, very improved, and it is possible that within a couple of years they may get another Nobel Prize. But the drawback of this computational method, they cannot predict the quaternary structures like hemoglobin. You have a tetramer of monomers, so it can only predict the monomeric structure. Tetrameric structure is still not very correct structures. Then ligand bound structures of protein is not that possible by the AlphaFold 2. So there are shortcomings of that. Cryoelectron microscopy, though very powerful technique, but in contrast to NMR you cannot go above 25,000. Same thing in cryoelectron microscopy, you cannot go below 80,000 daltons. So you require a bigger complex to determine the structures by cryoelectron microscope. But X-ray diffraction doesn't have that limitation. It can determine the structures of salt, NaCl, and it can also determine the structures as big as virus. So that is why it is becoming a very powerful method. The problem is you have to get the crystal of that macromolecules. Other low resolution structural methods called as one popular method that is not very commonly mentioned in the graduate textbooks, called as small angle scattering. Here you take a solution of proteins, solution of macromolecules, and the X-ray is scattered, not diffracted, scattered, and then you can predict the low resolution shape of the molecules, how they might be looking at in the solution. Fold into secondary structures. Secondary structures again properly fold into three-dimensional structures, and many three-dimensional structures comes close to form a quaternary structures. So in this category of secondary structure determination, if you have a protein and if you don't know the high resolution three-dimensional structure of that protein, one can get the protein in cuvette and determine the at least secondary structure of the protein using circular dichroism methods. It is an excellent method for determining the secondary structures, folding of the proteins, and it is basically done by because the molecule is somewhat CD sensitive, like helix, beta strand, and they absorbs the left or right-handed circularly polarized light. As a result, they produce ellipticity in the um, electromagnetic radiation. As a result, you can predict the secondary structures. For example, you can see in the right-hand side image, the alpha helical protein will show a typical CD pattern. If it is a beta sheet, then it will show something different pattern. And if the structure is random matlab there is no alpha helix, no beta sheet, or uh, there may be multiple conformation of proteins. It is a denatured proteins. You can say the omelette, where actually the protein is get denatured. Such proteins will show a typical CD spectrum. So for example, here I have shown the CD spectra of alpha helix, antiparallel beta sheets, disordered protein, collagen, which is in the order form, and collagen denatured, like gelatin, which is collagen denatured, which is being shown the CD pattern of that. And these are the different protein. For example, myoglobin protein on the right hand side, you can see the CD spectra. It is basically all alpha helical protein. Lactose dehydrogenase is another kind of mostly alpha helical with some beta strand proteins. Chymotrypsin are mainly a beta strand protein. So, and you can deconvolute this graph to get the how many alpha helix, how much percentage of alpha helix there, how much percentage of beta sheet there, how much percentage of random coils are there, and you can get the structures at the secondary structures level. So what you do in the macromolecular crystallography? So I told you now we focus on the X-ray crystallography. Now there are many terms which people use: X-ray crystallography, single crystal diffraction. Then people use XRD. People use macromolecular crystallography. This is basically a diffraction by X-ray. If we use a single crystal, it is called single crystal diffraction. So this macromolecular crystallography is nothing but a single crystal diffraction of a crystal which contain large macromolecules like protein, DNA or RNA. So these are called macromolecular crystallography. The XRD, X-ray diffraction, is basically a powder diffraction. For this macromolecular crystallography to determine the structures in the crystals. So you shine X-rays, monochromatic X-rays of particular wavelength, and you get a diffraction pattern. So these are dots. If you can see the circles of dots, you can see these are the diffraction patterns. And this diffraction pattern has all the information of the structures. So now it is really interesting, so that how we can get the structures from these dots to this electron density. And once we get the electron density, we can fit the atoms into that. And for example, here the electron density tryptophan amino acid is fitted, and above that there is a phenylalanine is fitted. So these are the different amino acids or atoms are fitted into electron density once we get the electron density. But that electron density has come from the diffraction image. How this is come? This is interesting. So I will explain into that also. And then once electron density you feed the atoms, then you can get the 3D structure. For example, here the structure of ribosome, which has millions of atoms into it, can be computationally fitted, and you can get the structures of ribosome. And the molecular weight is 56 daltons. On the other hand, a ribosome, for example, this is a eukaryotic ribosome, which is composed of RNA, protein, and these complex structures is 3.2 megadaltons. So 3200 kilodaltons size. This can be determined by the X-ray crystallography. Then photosystem 2, which composed of so many components of the proteins, different photoreceptors, metal centers, and different associated proteins that people have determined by isolating the photosystem from spinach leaves and got the crystal of that, and finally they have determined the structures of such a complex, which otherwise would not have been possible by other methods. And as a result, we now can how this photosystem works for splitting the water with the help of photons. Same thing, once we know the structures of ribosomes, we know many antibiotics which targets the ribosome and uh, perform the malfunctioning of the ribosome. As a result, the antibiotics works, and then specifically uh, so why X-rays? So you have been hearing the X-ray crystallography, macromolecular crystallography. Why we use X-rays? This is electromagnetic radiation. The wavelength and energy, they are inversely proportional. like more bigger wavelength, less energy, less wavelength, bigger energy. So in electromagnetic spectrum, if you can see, the X-ray has typically of order of keV, kilo electron volt, or wavelength of 1 angstrom, around that. So it can be 0.1 to 10 keV, 30 keV. These are X-rays. So these X-rays has a dimension of atoms. Like if you see the one uh, water structures, it is in the order of few angstroms. So this X-ray matches with the dimension of your molecules. As a result, one can very clearly probe the molecular structures. On the other hand, you have a gamma rays, which are very powerful. You might be using for the irradiation purpose, but they are not good for probing the structures of. So X-ray falls in the perfect energy level or wavelength range, so that can be used for the structural studies of molecules at molecular level. So by far the discovery of the X-rays is considered as one of the very important discovery. So the first Nobel Prize in 1901 or so was given to Roentgen for discovery of the X-rays. And then after that, Bragg's started determine investigated and found the structures of very small different small molecules, how NaCl are arranged in the salt, and then different structural studies of inorganic and then organic molecules has been done using the X-rays. Then why no X-ray microscope? So instead of diffraction, they should have been good. That it should have been X-ray microscope. We could have just put the protein solution and we could have seen the structure of protein under the microscope, but that is not possible. So in light microscope, you can see the cells, bacteria, and their movement by the visible light. But the X-rays cannot be done that way. Though you might have seen the electron microscopy. Electrons can be bent by the electromagnetic lenses, and one can get the high resolution structures, but not to that atomic level, but to a sufficiently high magnification by the electron focusing or electron microscopy. But the same thing cannot be done with the X-rays. As a result, we cannot study directly the molecules or atoms, how they are arranged in the molecules. So the primary reasons for that, the no lens, you cannot focus the X-rays like a lens. Just for magnetic lenses, you can focus the electrons. By normal optical lenses, you can focus the visible light, but by using the X-rays, uh, you cannot focus them by some kind of lenses. And the X-ray scattering from a single molecule is very weak. Suppose even if you keep some molecule inside some kind of microscope, the X-ray will scatter very less and you cannot really create an image out of that. So that's why the X-ray microscope is not present. So either you have visible microscope or you have electron microscope, but the X-ray microscope is not there. So how these X-rays are produced? So there are different methods to produce X-ray because you require intense beam of the X-rays. First is the lab source. Lab source, you have some kind of intensities of the X-rays. There are two parameters of the X-rays. I should tell you that one is the energy of X-rays or wavelength of X-rays. These are the same terms. And another is the brilliance of X-rays or flux of X-rays. So how many photons per unit time is we can call flux. And what kind of energy of that X-rays is a wavelength or energy of the X-rays. So this lab source produces certain kind of wavelength of X-rays or energy of the wavelength, but has very low flux, low brightness. Then you have a technique, you have the huge instrument facility called Synchrotron. Perimeter, one storage ring was there, and two proton with the TeV energy was collided, and they produce a God particle, and that got detected, and then that large hadron collider, you might heard about that. So it is similar to that. So synchrotron, what is there? That ring, you have electron is um, moving with the speed of close to the speed of light. Like light is in order of 10 raised to power 8 meter per second. And it is basically it is not exactly a circle, but it is a polygon of with many bend. This ring is, and at each bend you can see the X-ray is produced, and that X-rays are trapped to do some kind of experiment. These are called beam lines. So you can see the ring, which is a storage ring of synchrotron, and you can see something is coming out of that, that is X-rays coming out and focused in for experimental purpose. These are called beam lines. So this synchrotron produces an intense source of X-rays. And these are very, very intense. If you see on the right hand side, you see a graph and the year of development. So seal tube has a very low brilliance or flux of the X-ray. Number of photons is 10 to the power 10 per second. In the even less than that in the seal tube or rotating anode, which is lab source. The seal tube you also get in the airport. They produce X-ray by the seal tubes. And then you can see the bending magnet, wiggler and undulator, three words there. So these are basically different types of synchrotron radiation, or they are producing X-rays, but slightly different way in the synchrotron. One uses a bending magnet, other is a wiggler, and third is undulator. I come to that. And they produces very intense beams. For example, undulator produces 1,000 times more intense than bending magnet. Bending magnet itself produces 100 times more than the lab source. And free electron laser is even more intense than the undulator, and it basically very, very bright flux. Bright is the intensity of the light, intensity of the X-ray. So what is synchrotron radiation, which is more clearly shown here? It is the radiation emitted from an electron traveling at almost the speed of light. You can see the polygon of the ring, and that electron, which is shown in the here, where the electrons are moving at a close to almost speed of light, and at each bend the X-rays beamline radiation is produced, that is the magenta color arrow. And then there are different units at the beamline which produce the X-rays at the experimental station. These are called the synchrotron, typical synchrotron. You can see below there is a bending magnet source. So these are the bending magnet here. So electron bunch is moving. Actually, they are moving in the bunch, not like continuous manner. And once they deflected by the strong magnet, they produces X-rays, just like magenta arrows. But this is just one bend. Instead of one bend, if you have more bend, then it is called as a wiggler. Then you have even more intense X-rays. If the bending is very small and they overlap X-rays with each other constructively, then you produce even more, like 100-fold more intense than wiggler. So you have bending magnet, let us say it is produces 100 times more than the lab source. The wiggler in the synchrotron, which produces even 100-fold more than the these are the X-rays, they are produced and used in the experiment or experimental purpose of X-ray diffraction, X-ray scattering, and different use of this in the material as well as biological research. So what are the properties of synchrotron radiation? I've shown you. So they are high brilliance. They produce a very intense flux of photons. So 10 raised to power 12 photons per second. Then their energies are tunable. Like as I told you, in the lab source, you produce energy or wavelength of typical 1 or 2 maximum wavelength. But in synchrotron, you get a continuous spectrum of the X-ray radiation, and particular X-ray you can use for our experiment. Suppose it can be 1 angstrom, it can be 1.1 angstrom, it can be 1.5 angstrom, depending on the requirement. Then the high collimation. Collimation is a less divergence. So best collimated beam matlab it will not diverge, it will just go on parallel. So you get a better quality, better collimated beam by the synchrotron. So these are the three important properties which I told. I will not go into the polarization and fast time structure. And these good quality X-rays you get by the synchrotron you use for the X-ray crystallography. So in the RR-CAT Indore, we have two synchrotron located. One is called as Indus 1, other is called Indus 2 synchrotron. So Indus 1 synchrotron produces a soft X-rays like very high energy, very low energy or very high wavelength synchrotron, mostly ultraviolet and soft X-ray range. On the other hand, Indus 2, which are produced hard X-rays. Hard X-rays means less than 2 angstrom wavelength or energy of 10 keV kilo electron volt or so. So these are the two big facilities that RR-CAT Indore has, which is kind of BRC facility, and there are that beam lines surrounding this facility produces different centers for research called as beam lines. So he can see the picture of protein crystallography or macromolecular crystallography beam line. So you can see here the ring And this ring can be seen here. A lot of magnetic and other control units are there kept, and orange color which you are seeing here is a bending magnet. So at each bend you produces X-rays called as bending magnet radiation, which I told you. And this bending magnet radiation comes and we focus this into the experimental station using the beamline. So this is 36-meter long beamline, and in this part of this beamline we have the experimental station where we mount the crystal on sample mounted loop. The X-rays are coming from this side. Then there is a crystal mounted on this single axis goniometer, and simultaneously the crystals are cooled because of the prevent heating of the samples by X-rays. And here is some kind of microscope which is showing the crystal in the loop, and this crystal then diffract, and this diffraction is detected by called as the CCD detector. This is the largest detector India might have. It's a very costly detector, and you detect the diffraction by the X-ray diffraction by this detector. And here you can see diamond shape is the profile of the X-rays, which is very intense and which is seen by the X-ray cameras at this spot. And this is basically shined on the protein crystal, and you get a diffraction detected by CCD detector. And diffraction pattern I had shown you in my previous slide. So what do you do actually so there are back-end things for the crystallization. One need to have the purified protein for the crystallization. And this purification of the proteins, for this you require large amount of expressed protein. And that is by gene cloning in the expression vector. You take a gene, you clone into the expression vector, put into a bacteria, and bacteria produces protein of that gene. Either it is from human or it is from any frog or any plant. And this, once you have the mixture of proteins where one protein is overexpressed, you start purifying using column chromatography to get a purified protein. Once the purified protein is there, then you crystallize this protein. I will tell you the method of crystallization. Once that crystal is there, then you diffract on the X-rays and you get a diffraction pattern. You process the data, then solve the phase problem. I will come to that by these three different method: MIR, MAD, or MR. Just have remember these three words. I will come to that. And you get electron density. You feed the model into that and validate the structures using some other methods. And finally, you get the structures of protein which you submit in the protein data bank. And then the researcher use these protein structures for analyzing the function of this protein. In large quantity. For example, typically you require 10 to 100 milligram protein in a concentrated form. For example, 10 milligram per ml to say 50 milligram per ml, and you require these solutions at least 500 microliter or so. So different method is vapor diffusion method, batch method, dialysis method, interface diffusion method. So what you do is you take some kind of well Into that you put a crystallization solution, and on the cover slip you put a protein solution as well as this crystallization solution 1:1, and store that at constant temperature for maybe week or 15 days, and you get crystal appeared in the drop. So one has to try hundreds of different crystallization conditions. Each condition has a different pH, different salt, different additives. So you don't know beforehand which solutions will give the particular protein crystals. So you require to try different crystallization conditions. So this is what happened in this case. When you put the protein drop in the reservoir, it slowly get concentrated, and if it is a favorable condition that it may crystallizes out. And same thing, you have a sitting drop vapor diffusion technique. Instead of hanging on the cover slide, you can put in some kind of sitting arrangement. Then in this micro batch under oil, you put this protein 1 and crystallization solution 1 drop inside the oil, which is immiscible with the drop. And as this drop slowly evaporates, the crystal might appear there. Then there is a dialysis, then pre-interface diffusion. These are the different methods of the crystallization. Nowadays, people using a crystallization robot for setting up thousands of conditions. So robots is very handy for that, but one can do it by the hand also. You might be asking the questions, one protein is form a crystal. Are they active? Yes. Proteins are joined together to form the crystal, but they have not lost activity. That is shown by this picture. This is a phosphatase N protein which got a crystalline, and I soak the substrate of this enzyme phosphatase in the crystal, and it start producing the product, which is a colored product. So you can see the blue color of the crystal is due to the production of the product inside the crystal. So though it is packed in the crystal lattice, it is still the active protein. So proteins are active in the crystals, and that is why the structures which we get using protein crystallography are active structures. It's not that denatured or something like that. It is very well biological structures. So what this protein get packed to form the lattice, periodic lattice. We call that unit cell. It has a dimension of A, B, C, and alpha, beta, gamma. So these are called the repeating unit of the protein crystals. So you can see the repeating unit, and this repeating unit get repeated and form the crystals. And from there, how this diffraction occurs, I will tell you. So this is a repeating unit, and one or two molecules or more molecules can be there in the one unit cell. This is just to show that how these are packed. So there are, I will just skip this, 65 different way the protein molecules or macromolecules can pack with a different symmetry. I will not go into the detail of that, but you just remember the 65 different way the protein packed into the crystals, and this crystal we use for diffraction. So this is the some kind of mounting device, and at the tip of this you can see the loop, and in the loop you can see the crystal which is there mounted using the microscope. And the other scattered beam you get on the detector. But what people do is they don't just do the crystal and mount it. Before that they freeze the crystal in the liquid nitrogen or liquid ethane or liquid propane, and these frozen crystals are mounted on the goniometer. So this is diffraction patterns. As I told you, in the center there is a direct beam stop, and you get a diffraction pattern. So dots like these patterns. So what you do is you take one snap, you rotate the crystal for 1 degree, then take the small spot, then again rotate from 1 to 2 degree, take another spot, then rotate 3 to 4 degree, then take another spot, 4 to 5 another spots, and as a result you can create diffraction images in the order of 360 or whole rotations. And these diffraction patterns now used for solving the protein structures. Sometimes what happens, the crystals are not perfectly packed. They have the mosaicity. So like all bricks are not arranged regularly perfectly. There is a gaps or there is a tilt in the bricks and all those things. So that gives a weird diffraction patterns, and it is very difficult to process this kind of data to get the diffraction pattern. So you want the lower one, which is shown a good diffraction pattern. This is bad diffraction pattern because of the defects in the crystals. So what you do is here, so you have a crystal. This is a crystal. This is a schematic of the crystals where the real molecules are packed in a lattice. Okay. Then you produce X-rays, you shine X-rays on them, and you produces diffraction patterns. May I know, organizer, how much time do you have?
Sir, it is up to 3 p.m.
Up to 3 o'clock.
3 o'clock.
Okay. So you produce a diffraction patterns, which is basically what happen in this diffraction pattern. If you can see closely, there is a different intensities. One spots are light, other spots are very dark, like that. Okay, and they are arranged in a particular order. So this is we call as a reciprocal space. What is reciprocal space? This is one kind of imaginary space. So when you shine X-rays, you get a diffraction pattern. So this molecule get transformed. This content of this molecule is reflected in a reciprocal space. This is called as a diffraction pattern. So if you have more dimension on this axis, then you have less dimension on this axis in the reciprocal space. Everything get reciprocal. Let us say you have unit cell ABC here. So this unit cell get reversed in the reciprocal space. That's why it is called as reciprocal space. And this FT is Fourier transform. This is called as a Fourier transform. So from real space to reciprocal space, and then you do the calculations and get the electron density by the inverse Fourier, and again get the real space. Real space is atom where you can see the atoms. Now, what is there in this reciprocal space? So each spot is basically a reflection called as a X-ray diffraction spot or reflections. This reflection has a particular intensity and also a phase angle. Like phase, you can see there is a phase shift in the electromagnetic radiation, which can be from 0 to 360. So which can assign each spot can have the different phase angles. So each spot will have different intensities as well as different phase angles. If we know both of them, then we can compute the inverse Fourier and we can get the electron density where we can feed the model and you can get the structures. So the simplistic explanation of this diffraction and Fourier patterns and all those things is explained by the Bragg's law. What Bragg's law say? N lambda is equal to 2d sine theta. So when this X-rays fall, it is basically some kind of imaginary planes. This is a unit cell and you can see the imaginary planes there. So these imaginary planes and if the angle of theta is such that it corresponds to some kind of integral value of the lambda, then you can get a Bragg spot, that is a diffraction spot. So you have a single crystal, you have the periodicity in them, and you have a set of imaginary planes cutting this lattice. This is called imaginary set of planes, and this imaginary set of planes, if we shine the X-rays and if your theta angle, the angle incident on these imaginary planes is matching with some kind of spacing between them, d spacing, then you produce integral values of this of lambda. Let us say 1 angstrom, 2 angstrom, 3 angstrom integral value. Then you produce as a spot. Then this reflections they get overlap and produces a diffraction pattern. So as I told you, this diffraction pattern is nothing but some kind of lattice points which you are projecting them on the detector. As a result, you are getting the diffraction pattern like this. So this is a three-dimensional lattice, but because our detectors are not circular or other things, we have two-dimensional detector. So basically you are catching the diffraction pattern or diffraction lattice points on this two-dimensional space by certain conditions called Ewald's pair of satisfaction of the Bragg's law. I will not go into the details, I will just skip this and then. So each spots, diffraction spots, is a reflection from a set of planes. So like this spot, diffraction spot, is a reflection from particular set of planes. This is from different set of planes. This is from different set of planes. And how much electrons content on the set of planes determine the intensity of these spots. And the positions of these electrons with respect to each other determine the phase angle of this spot. So these spots can be 0° phase angle, this can be 20°, this can be 10°, this can be 110°, whatever. So this one can arrange, we can get the diffraction patterns, their intensities, their phase angles, and we can get the electron density. So now the concept of atomic scattering factors, I will just briefly touch on them because unless I mention this concept, you cannot really do the justice with this crystallography. So as I told you that atoms have the electrons, spherical electrons around that, which is in the order of your wavelength. As I told you earlier, when X-rays shine on this and you produces a diffraction pattern. This is just from single atom diffraction pattern. So this diffraction pattern, this Gaussian diffraction pattern is nothing but a Fourier transform of electron density. So electron density here you can see, and the Fourier transform of that is the structure factors. So for example, at 0 degree, this corresponds to number of electrons present in the particular atoms. For example, iron. Iron, the number of atoms, number of electrons are 26. So you have atomic scattering is 26 at 0 degree angle. It is more here. As you go away from this, your diffraction, atomic scattering decreases, and this scattering factor is basically used to draw the intensities. So what is this scattering factor? So as I told you, structure factor. This is nothing but has come from the each atom will have atomic scattering factor depending on their positions and intensities of electrons. Same thing is diffraction factor, we can call as a structure factor. What is the structure factor? The structure factor is nothing but a convolution of different structure factor, different atomic scattering factor of the atoms. So this waves is, let us say, from carbon atom located at particular spot. This wave is located at some other spot of the from oxygen atom, like that. As a convolution of atom, you produces the intensity of the structure factor. So every atom in the unit cell contribute to every reflection in the diffraction patterns. What that means? That means the spot which you are seeing in the diffraction space has a reflection from each and every atom of the unit cell or crystal lattice. Each one contributes towards them and produces structure factors. And structure factor, this is called FHKL. Just remember that it is denoted as FHKL, is a wave created by the superposition of many individual waves, that is atomic scattering factor, each resulted from individual atoms. So I will just summarize here that this structure factor is nothing but a diffraction, say one spot in the diffraction pattern is nothing but the contribution from all atoms of the unit cell depending on their positions and intensities. As a result, you produce a diffraction pattern. So what do you do is you produce a electron density. Electron density is nothing but summation. This is the summation sign of all structure factor here, with their phase angles. This is the phase angles, and this phase angle you cannot determine directly by the detector. You have to get it indirectly, and that is called as a phase problem that I told you, crystallographic phase problem. So this alpha HKL, you get the structure factor amplitude or intensities of the structure factor by this diffraction pattern, but not the phase angle. So phase angle of each spot is determined indirectly, and this is very popularly called as a crystallographic phase problem. So these are the phase problem by the different method. One is a molecular replacement. If we know the homologous structure, let us say you know the suppose you want to find out the structure of hemoglobin of some dog, which is say 70% similar to, let us say, some fish hemoglobin. So you can use the fish hemoglobin to get the structure of dog hemoglobin by some kind of technique called molecular replacement, which provided the approximate phase angles to these diffraction spots. Then there is a called as multiple isomorphous replacement. There is heavy atoms which are soaked and you get diffraction patterns. As a result, your diffraction pattern get perturbed and you have a uh, uh, solution to the phase problem. In these days, because of the synchrotron, you can do the single wavelength and multi-wavelength anomalous dispersion. So what it means actually, you can change the energy of the X-rays and as a result, you can get the diffraction solution to the phase problem. For example, this is the two diffraction pattern which is shown here. One is a native without heavy atoms, other with the heavy atom, and you can see the change in electron density, change in the diffraction intensity and the underlying one. And this because you added the heavy atom in the crystals, as a result you see the difference in the intensities, and this difference in the intensities are exploited to get the position of the heavy atoms, and this heavy atom position you can get somehow the phase angles of each reflections. I will not go into the details of that, but just to remember here that once we have the heavy atom soaked, you have the diffraction pattern of native crystals as well as diffraction patterns of heavy atoms soaked into the crystal, and comparing these two diffraction pattern you can get the phase angles of each reflections. I will skip this slide. So each diffraction pattern you have this is called as the intensity 228 for each set of each plane. This is plane called that imaginary plane, set of planes 200. So this is a reflection from, let us say, 200 plane, and you have intensity of 228 and phase angle of 180. Another set of plane 101, you have intensity of 10, very less, and phase angle of 90. All this data you collect using computational and X-ray diffraction, and then by putting in this electron density you can get the electron density map. To further improve this electron density map as of computational methods, and then start fitting the atoms into this. So for example, here there is electron density, then you feed the atoms and you get the where are my atoms and ligands bound in the protein structures. So one has to be very important of the Ramachandran plot. You must have heard about that Ramachandran plot. It is the torsion angle phi and psi. The ratio of that phi and psi should be particular for right helical, alpha helical regions, and beta region will lie into this. And these your structures which you predicted uh, based on the electron density should match this Ramachandran plot. You should not deviate from the Ramachandran plot. So that is the first important criteria to validate your structures. There is another factor called R factor, reliability factor or residual factor, which tells us how much is best your structure is. For example, zero point 0.2 it is good, 0.4 it is manageable, and if it is 0.5 or 0.6 R factor, they are very bad structures and one cannot believe the model of the structures. So these are the different parameters of the validity of the structures. The above 4DI, this is the protein data bank entry number. The above structure is quite good structures, and the below one structure is very bad structures, uh, but nevertheless submitted because that is the best you can get from the crystals. But the reliability of the structures you can predict by the validation, and you can use depending on if it is good structures. I know the confidence in my structures. If it is bad structures, then I know the confidence is very low in that structure. So resolution of the structure is a very important area. So for example, you must have read about this structure is solved at 1 angstrom resolution, this structure is solved at 2 angstrom resolution, and this is very low resolution. So you can see in 1 angstrom each atom you can see separately. For example, this phenylalanine, and you can see the all 6 carbons are located nicely, but the resolution is poor, then it will just produce as a blob of that electron density. And the resolution is very poor, then you can just treat the side chains of the amino acid with a low confidence. These are the different representation of the protein structures. Once we have the atomic model, you can present the structure in the cartoon form, you can present the structure in the stick form, or in the ball form depending on the van der Waals radius of each atoms, and the surface form. So these are especially ball and surface form and stick form. They are very complex to understand. So most often if you have seen your biochemistry books or something, there mostly it is given in the cartoon form, which is the most simplistic form of representing the protein structure. But more realistic structures, which will, if you have the X-ray microscope, it will more look like the surface or balls, mostly surface like. So what are the problems in X-ray crystallography? So why not all structures are known? Yes, because these multi-protein complexes, for example, this nucleosome and different proteins bound to these nucleosomes, such structures which are multi-components. 7, 8, 10 proteins are bound to DNA, and DNA is very big. Such kind of big structures, they form crystals very in a difficult way. We have to engineer the protein. We have to try different conditions to get the crystal. So many of these big multi-protein complexes, DNA, RNA complexes, DNA protein complexes, RNA protein complexes, they don't form the crystal very easily. So that is a big problem for X-ray crystallography to determine their structures. The membrane protein. The membrane protein are very sticky because they have hydrophobic regions surrounding them. As a result, they don't form the crystal very easily. So they have a particular method to crystallize. So very difficult area to form the protein crystals. Then some proteins are the flexible proteins. For example, you can see the structure of these proteins. Their N-terminal end or C-terminal end are very flexible. Some loops are flexible. They also don't form the crystals very easily. And very large unit cell. For example, you have the ribosome structures, and their unit cell, which is repeating cell, can be very big. And such structures are very difficult to solve because you have very crowded space in the reciprocal. As I told you, you have all the reciprocal things. If you have big unit cell, then all your spots are very close to each other. If you have small unit cell, your spots are quite distant from each other and very easy to solve. But if your structure with a very large unit cell, then your diffraction patterns are very crowded and difficult to solve the structures. So this all things I have taken from the biomolecular crystallography Bernard Root, crystallography made crystal clear, guide for user macromolecular models by Gale Rhodes, and principle of protein X-ray crystallography by Drenth. So I have taken all the information from there. Thank you. Any questions? Dr. Nitin, any questions?
Yes, I'm waiting for the audience to ask the questions. Hello, dear colleagues. Do you have any questions? Hello?
No, sir.
Hello?
No questions from my side.
It seems that physics cannot be easily digested by biology people.
Yes, sir. Yes, that's true.
No, this is the best effort by me as a biologist to, this is the, if I digested further, then there will be no macromolecular crystallography.
Hello, sir.
Yes.
Sir, I don't have any type of the query, but I'm very excited to know what was the turning point for you that have shifted you from botanist to become a physicist.
Yeah, actually, I joined BRC. There is a process for joining as a training school. So we join through that, and then vacancy comes depending on your, let us say you have 5 seats available. So this topper gets to choose everything. So I didn't have choice to choose the botany because botany was not there anyway. So I found the crystallography was best among the available, and these are really you can get the basic of this. And for example, even plant proteins, they are very unique, and if you know the structures of them, we can help to understand the function. So I knew that this area is a very fertile area, so I just jumped upon that. But
Yeah, I will add one point from my side in favor of Dr. Ravindra Makde.
Yes,
When
sir.
he was doing MS botany, at that time, the research students from physics and research students from mathematics were frightened of him because he was knowing more mathematics and more physics than the students of MSc physics and MSc mathematics.
It is so kind word. Thank you.
Yeah, this was the fact at that time. I still remember many of the PhD students of mathematics and physics were frightened of you.
That's why I'm asking, na, sir.
Yes, yes,
That's
yes.
why I'm asking.
Yeah, he had
That
got
means,
so much interest in
Actually,
physics at that time.
I will tell you one thing. Actually, this is very easy subject. If you start practicing this subject, you will find it much easier than conceptually. Like, you don't have to understand each and every atomic details because there are computer processing, and that physicist made your life quite easier. So it is not so difficult area for biologists. Many biologists now coming into this area. What is more important is the problem, which problem you are attacking. For example, you have a protein from some seeds, and which is very important. So that is most important. Even if you don't know technique, you can go to some crystallographer and ask him, can you get the structures, why this protein is so peculiar, then that is most important. And that will fetch you good paper, not just solving the structures.
So if there are no questions, I on behalf of Rashtrasanta Tukaroji Maharaj Nagpur University and UGC HRDC Center Nagpur is thankful to Dr. Ravindra Makde to introduce the aspects of crystallography to us. Though we are the students, I will consider ourselves as students because a teacher is always a learner. He is always student throughout his life. So we, being the students of biology, have learned so many things of about the physics from you. So I'm thankful to you, Dr. Ravindra.
I'm
Thank you very
thankful
much
for
once
giving
again.
me opportunity.
Yeah,
Thank you.
it was our
Thank you,
pleasure
sir.
to have you as a guest speaker.
Thank you. I will log off myself.
Thank you. Thank you so much, sir.
Thank you.
Thank you all of you. सर काही instructions आहेत का डोंगरवार सर? Friends, I have shared a timetable of seminar.
हाँ,
Kindly
madam.
go through it. Yes, seminarचा timetable मी पाठवला आहे. You just go through it. And I have shared one flyer regarding one webinar on 12th of September on Monday. मंडे आपका शेड्यूल का टाइमिंग मैंने बताया है. योगा सेशन आपका सुबह 7 to 8 रहेगा. रिमेनिंग टाइमिंग एज इट इज. एंड 3 to 4 सेमिनार का एक स्लॉट रहेगा. 4 पार्टिसिपेंट्स के सेमिनार रहेंगे क्योंकि 1 घंटे का ही स्लॉट अपने पास है, क्योंकि 4 to 5 वेबिनार इज देयर. तो वेबिनार के लिंक और सारी चीज़ें आपको मेल पे शेयर होगी. और अपना जो सेशन है वो 4 बजे तक या तो 3:45 तक अपन एंड करने की कोशिश करेंगे. सेमिनार जो अपने 4, अभी मैं फिर से बता रही हूँ, जो आप सेमिनार प्रेजेंट करेंगे वो प्लीज़ ज़्यादा लेंदी मत करिए. आपका कंप्लीट सेमिनार एक्सप्लेन होके 10 मिनट में होना चाहिए. तो बहुत ज़्यादा इलेबोरेटिव नहीं, जस्ट आपके जो कंसेप्ट सिर्फ एक्सप्लेन करना है, मेजर फाइंडिंग्स आपको एक्सप्लेन करनी है. 8 to 10 मिनट्स में आपका सेमिनार खत्म होना चाहिए. अगर किसी को कुछ क्वेश्चन आंसर्स है तो 2-3 मिनट और लगेंगे. सो कुछ क्वेरी होगी, कुछ पूछना होगा तो पूछिए. Otherwise, I'll end the meeting and see you on Monday at 6:45, and our session will start from 7 o'clock.
Okay, madam. Okay, thank you.
Thank you. Any doubt?
No, madam.
Okay, ठीक है. Thank you all of you.
Thank you.